I'm honored to present our study, Cross-Continental Transmission and Host Adaptation of Mycobacterium tuberculosis in China, unveiled by population history reconstruction and adaptive evolution signal detection. Tuberculosis is a respiratory infection. In 2023, it caused over 1.25 million deaths. This pathogen, Mycobacterium tuberculosis complex, MTBC, is an obligate human pathogen, and its spread and evolution are tightly linked to host populations. MTBC comprises lineages, yet China shows unusually low lineage diversity. We propose two hypotheses. Recent L2 and L4 introductions replaced ancient strains, or a few strains underwent host adaptation to Chinese populations. We find 4 MTBC sub-lineages entered China 1,000 to 400 years ago from Europe to China, seeded local clades, and underwent 3 population expansions. We reveal convergent evolution in local clades, consistent with selection pressures from host macrophages. We also introduce new bioinformatics methods, estimate selection pressures by neutral mutation simulation, and detect adaptive signals by DNA information entropy. China's endemic MTBC is dominated by lineage L2 and L4. These strains diversified about 1,000 to 400 years ago and cluster into local clades whose internal diversity exceeds that of their sister clades. Notably, L4.5 is almost restricted to China. 4 sublineages formed early spread between South China and Western Europe 600 to 300 years ago. In the last 200 years, both regions became global hubs. L2 likely arose in Fujian and Anhui, moved to Europe, then spread globally. L4.2 and L4.4 likely originated in Western Europe, entered China and diffused outward. L4.5 likely originated in Zhuochong and spread mainly domestically. Geographic isolation and limited maritime corridors may have constrained lineage diversity in China. We identify 3 MTBC population expansions in China that align with regional cold periods, coincide with post-late Ming and Taiping rebellion human population rebounds. Low temperatures likely facilitated transmission, while weakened post-war infrastructure and human population growth together may accelerate MTBC expansion. 300 to 200 years ago, local MTBC clades in China show elevated mutation rates and stronger positive selection on macrophage invasion genes and T-cell epitopes. L4.5 shows host adaptation about 600 years ago, other clades about 250 to 200 years ago. These adaptations likely promoted local expansion and maintained low lineage diversity. Information entropy analyses highlight 12 genes with strong adaptive signals across local clades in China. RV0801 shows the strongest signal. Infection assays demonstrate RV0801 enhanced intracellular survival in macrophages. Protein is predicted as glyoxylase, which detoxifies methylglyoxyl, boosting resistance to macrophage. These findings indicate macrophage-driven selection. Local clades in China share small indels enriched in PE-PGRS family genes, concentrated in the PG2 helix. In PE-PGRS3, frame shifts from indels truncate the long C-terminus, altering macrophage adhesion and immune evasion. Macrophage and humoral immune pressures appear to shape adaptive evolution in these clades. We detect 5 clade-specific large deletions in local clades. RDA removes the gene RV0576. RDB to E occur in PE-PGRS genes, deleting the PG2 helix and long C-terminus. These changes reduce TNF-alpha induction and modify macrophage adhesion, aiding immune evasion. Such clade-specific deletions further support adaptation under macrophage selection pressure. This study reconstructed MTBC's history in China, showing repeated transcontinental introductions followed by local adaptation via an integrated framework, lineage dynamics, historical mutations, DNA information entropy, and structural variation analyses. New methods was developed to quantify selection pressures and identify the genes undergoing evolution. Infections validate RV0801 as boosting survival in human macrophages. And geographic isolation, war, and temperature cycles likely drove macrophage-targeted adaptations and dominance of local clades, revealing regional pathogen-host coevolution's global impact and offering a model to reconstruct spread and predict local adaptation.